as the pandemic has shown us, it is not the virus, it is how our immune system reacts to the virus that decides whether we get an asymptomatic infection or we become serious or if people die. We are all alive and participating in this seminar today because of our immune systems. So it's a fascinating system, and I'll try to give an overview in 20 minutes, an introduction to how it works, because the details are amazingly deep, and it's just not possible to cover details in 20 minutes or even 20 days. Let's start with your hypothetical villain or a virus. So we all know what the virus wants to do, right? A virus wants to fix to the cell wall, enter inside with the genes and insert the genome into the cell and take over the functioning of the cell to make its own copies. Okay, that's what the virus wants to do. So who do you think is the primary, which cell or which antibody or what is it that primarily defends your cell against the virus? It is not any part of the immune system, but every human cell has its own defenses against viruses. So each of our cells is like a samurai warrior. You know what is the Japanese tradition of samurai warrior? That the warrior will fight, and if the warrior is defeated, he will commit suicide. He will not become a slave. He will not become a prisoner of war. So human cells are like that. Every human cell tries to fight back against the virus which is trying to enter. And if it fails, and then it becomes a slave to the virus, it tries to commit suicide. The first line of defense, every cell basically detects that a virus has entered. Supposing this is an RNA virus, it enters into the cell, there are certain receptors inside the cell which detect that the virus or a strange RNA or a DNA has entered into the cell. Then the signal is passed on to the nucleus and certain genes are activated. What do? They start making interferon. Okay? This interferon is then spread around the cell, and the other neighboring cells are alerted that a virus attack is going to come. Then interferon acts on those cells, signals the nucleus to start making antiviral proteins. This is the first line of defense against viruses. Interferon has many actions. It generates the synthesis of viperin, tetherine, which are antiviral proteins. It tries to shut down the protein synthesis. Why? Because if you allow protein synthesis to continue, then the cells are going to make viral proteins, which you don't want. It will reduce viral replication, activates antiviral enzymes, and it is a powerful signal to call other immune cells for the fight. So interferon is a good signal for T cells, natural killer cells, to come and kill the infected cells. Once a cell is infected by a virus, you don't want that cell to be alive. You want to kill that cell, otherwise it will simply make more and more copies of the virus. So as we showed that RNA receptor, what is that receptor which detects that the virus has come? There are many such receptors, and they are known as PRR or pattern recognition receptors. Some of the receptors are on the surface of the cell. Okay, so this is a virus. It will attach to this receptor and the signal goes into the nucleus, and interferon and other cytokines are manufactured. Whereas some of the pattern recognition receptors are inside the cell. So the virus is already entered inside. It will detect and give the signal to the nucleus. One of the examples is TLR or toll-like receptors. There are many such, there are at least 10 TLRs. Some TLRs are sitting on the surface of the cell. As you can see, if this is the cell, these are on the surface, TLR2, TLR6, TLR1. They detect parasite antigens, fungal antigens, bacterial lipopolysaccharide or flagella antigens, and they give a signal inside. Whereas some of the TLRs are inside the cell and they will detect DNA virus, RNA viruses or bacterial DNA fragments. So by many of these signals, ultimately the signal goes to the nucleus so that the cell can fight back. So TLR is just one system. There are many other systems like RIG-like receptors known as RLR or NOD-like receptors known as NLR. And some of these receptors, like especially NLR, when it is detecting the entry of a pathogen, it becomes transformed into what is called an inflammasome, and that can lead to the synthesis of enzymes like caspase, which commit suicide basically. So what happens when a stimulus like an entry of a pathogen is detected? The stimulated cell, even normal human cells, not immune cells, all human cells can produce cytokines. And these cytokines, interferon is also one type of cytokine. So they will go and stimulate other cells to fight back. Okay? But there are some cells which are very, very good at making cytokines, especially helper T cells. They are champions at producing lots of range of cytokines. And even macrophages or B cells, they are all good at producing cytokines. So these are examples of various cytokines. Interferon, chemokine family. Chemokine means they attract other cells for the fight. So they will stimulate the migration of neutrophils, macrophages and other phagocytes to the site of action. Tumor necrosis factor, TNF. These are stimulatory, inflammatory cytokines. Interleukin family, as we know, we have so many interleukins, and now we have drugs to antagonize the unwanted interleukins, etc. Then hematopoietins like GCSF or colony stimulating factors, they are also a type of cytokines to promote cell proliferation, and TGF family. So they are all examples of cytokines which are released at the time of an attack. The next component of the innate immune system that we are talking about is complement family. What is complement? It is a system of 20 proteins that are present in high concentration in our blood. And this is a very old system as far as evolution is concerned. The old 700 million years ago sea urchins, they also had it. And the adaptive immune system is a new evolutionary new system comparatively. This system is ready to go at birth. Basically, it contains so many different proteins, and you know coagulation cascade, right? When one clotting factor is activated, it will stimulate the next one, the next one, next one till a clot is formed. So complement is nothing different. If a complement 3 is first activated or broken down into 3 and 3b, it will start stimulating the next complement factors. And the last ones to be stimulated are 6, 7, 8, 9. Complement 6, 7, 8, 9. They together, when they are activated, they form a deadly complex called a membrane attack complex, which can punch a hole into the bacterium or into the capsule of a virus or into an infected cell to kill that. Okay? So, how is this complement activated? There are 3 pathways. Let's take this alternative pathway first. What is alternative or spontaneous pathway? So it is like a magic bullet. Supposing all of you sitting here, you are normal cells, but among you there are some bacteria or there are 1 or 2 viruses. So if I take a machine gun and start shooting all over, okay, and I have a magic machine gun, that it will not damage the normal cells but only kill the virus or the bacteria. So that is something like the alternative pathway. This complement 3 is spontaneously getting activated in the blood randomly, and it will generate membrane attack complex and kill the bacteria, but it cannot harm the normal cells if they are healthy because of presence of certain protective enzymes on their cell. Okay, this is called an alternative pathway. Classical pathway, when complement gets activated, whenever it comes across any antigen antibody complexes. So if you have antibodies which are sticking to the surface of a bacterium or a virus, this complex is a good target for complement activation. So it is a wonderful system that the complement will go and attack wherever there is an antigen antibody complex and kill that particular pathogen. And another pathway is the lectin pathway, where mannose, which is one of the structures on the surface of some bacteria, acts as a trigger for complement activation. So whatever way, if you activate complement, the first one is C3, which is broken down to C3A and C3B. And this further increases the process by a self-amplification loop, C5 breaks down into C5A, C5B, and then this stimulates C6, 7, 8, 9, forms the membrane attack complex to kill the infected cell or a bacterium. Even this complement 3A or 5A, they are not wasted. They are also like chemokines to attract more neutrophils and monocytes to the fight. Which are the cells of the immune system? The innate immune system, the first one to enter the fight is always macrophage. You know, macrophages are there in the lungs, under the skin. Wherever there is a possible point of entry for a bacterium, macrophage is the first one that will defend it. Normally they are quiet, they are doing a job like a garbage collector. Because in our body, 1 million body cells die every second. So all that debris or garbage has to be cleared up, and who is doing that? Macrophages. They keep on clearing the garbage. But when cells are dying, not because of natural death, but because of inflammation or damage or trauma or infection, then they pick up the signals and become activated. So when they become activated, they become warriors. Now this macrophage, which is doing the job of garbage collection, it detects the presence of interferon or lipopolysaccharide from a bacterium, it becomes angry. Now it's an activated macrophage, it will start making tumor necrosis factor, TNF, from itself. It will start displaying these molecules on the surface, MHC, okay? MHC2 molecules on the surface. What is the job of that? We'll come to later. So these macrophages will then stimulate the natural killer cells via interleukin 12, via TNF, and it will stimulate the macrophage back by interferon, and especially if the macrophage detects lipopolysaccharide which has come from a bacterium, it gets even more activated, and the process of activating all these phagocytes will start. Soon these signals will attract neutrophils to join the fight. Neutrophils are professional killers or phagocytes. Of course, we are very much in awe about T cells and B cells, which are fancy cells. But we have to understand that without neutrophils, human being cannot survive more than 5 to 6 days. Without B cells or T cells, human being can still survive for weeks. So these are major fighter cells which reach the infected tissue within 30 minutes. They have got receptor on the surface for FC component of the antibody for complement, and they release all these deadly chemicals, and these are so powerful chemicals that they destroy the bacteria and the nearby healthy cells to create pus over there. Even when they die, they release these structures called neutrophil extracellular traps, NET, and these traps also catch microbes and kill them. So these are all, in short, the components of the innate immune system, which are always ready to fight. But when the infections are more severe and the innate system cannot take care, then we have the adaptive immune system, which is later on in evolution. The lowest animals don't have this, but we have this. Now you will be surprised to know that let there be any virus, let there be artificially created virus, or mutant virus, or mutant bacteria, or fungus, or whatever it is, we already have antibodies against anything that nature or humans can throw at us. Okay, they are already there in us. Where are those antibodies? If you try to go and measure antibodies in blood from somebody who has never been infected or vaccinated against COVID, you will not get COVID antibodies. So where are those? They are sitting here on the surface of the B cells which make them. Okay, so how come we have antibodies against anything that can even arise in future? Because ultimately all bacteria, viruses, fungi, they have on their surface organic molecules, and there are many permutations and combinations of amino acids or lipids or carbohydrates, but they are not infinite. So an estimate says that we only need about 100 million different antibodies to take care of any pathogen that can ever arise. And we have enough B cells, different B cells, which can produce each one of these 100 million different antibodies to attach with the pathogen. So these they need to be recognized, and we have 3 billion B cells. So for each possible antigen we have 30 B cells available, and we have 300 billion T cells. So these make the antibody. So these antibodies are sitting on the surface of the B cells. Okay, we know the structure of the antibody. This is the FAB region, this is the FC region, and whenever, now this is the antibody sitting on the surface of the B cell which has made it, and if it comes in contact with the antigen, it will get activated. Where does this contact happen? This happens in the lymph node. Now the lymph which carries the complement bound antigen enters the node. B cells go into the cortex of the lymph node. There they meet these follicular dendritic cell, FDC. B cell finds the antigen, multiplies, and there is a further stimulation and maturation of the B cell with help of the T cell. What does antibody do? This is the virus, this is the antibody which is neutralizing the virus, or it can opsonize the virus. That is the virus, the antibody is attached to that. The other end of the antibody, which is known as FC, it is recognized by this phagocyte, which will go and eat this virus. Or so this is a macrophage which has a receptor for this FC part of the antibody, and the antibody other end is attaching with the antigen. This is an infected cell and it is expressing the virus on its surface. So the antibody acts like a bridge to call the macrophage to attack the infected cell. Or it can attract complement, as we saw before. Complement comes where there is an antigen-antibody complex and attacks this cell, create this membrane attack complex. Or a killer T cell may come and attack this infected cell. So antibodies themselves do not kill, but they will neutralize or they will call upon other cells. So virgin B cell activated to fight back, it will become a plasma cell and make 2000 antibody molecules per second. For 7 days it will live and then it will die. But some cells become long-lived or memory cell, and when there is a second attack they will get activated. How do T cells get activated? In the tissues there are these dendritic cells. They sense or they eat the bacteria or the virus, and a part of the virus is expressed on the surface of the cell, and it is like a notice board. MHC2 molecule acts like a notice board, and in that notice board a part of the virus is digested and displayed, that look, this is what I have captured. Then this dendritic cell will go to the lymph node where it will meet the helper T cell. Now there are 300 billion helper T cells which are circulating. When the right T cell meets the dendritic cell, it will get activated. And what will the activated helper T cell do? It is a cytokine factory. It will, so this is the antigen presenting cell. It will pick up the antigen, show it to the T cell. The T helper cell then goes and helps the B cell to make antibodies, goes and helps the macrophages to fight, and goes and helps the other killer T cells to fight. Now there is another type of presentation. The first presentation we saw was the dendritic cell captured the virus, digested it, broke it into pieces, put it up on a notice board. What notice board? MHC2 molecule is the notice board. That is called type 2 presentation. Now type 1 presentation is every cell in the body, every day it is required to show what it is doing for 24 hours. Every cell in the body is making some protein through the day. Okay? Whatever protein it makes, it chops up a bit of it and displays on the surface on MHC1 molecule that this is work I have done today. So supposing a virus has entered the cell, that cell will make virus proteins, and at the end of the day it will show, see, I am making viral proteins. So this is detected by the killer T cells. Okay? So this is known as the type 1 presentation. So the infected cells are detected by killer T cells. How do killer T cells get activated? I'll just take a couple of minutes more. So when the killer T cell comes to know that a particular cell is infected, it will attack that cell by using either perforin system or FASL system, and some of the killer T cells become memory cells. So this is the biology of the killer cells. T cells are activated. Most of them die after they have got rid of the infection. Some of them live in the tissues as memory T cells. Some live in the central bone marrow or the lymph nodes as central memory T cells, which are ready to get activated on the second attack. So this is in short a summary of the entire immune system. I'll just go through fast. In the second infection, all these systems are accelerated. So if you are vaccinated or you have been infected once, the activation of all these systems takes place much faster. But switching off the immune system is also important. Otherwise, you will get damage to the own body. So as the antigen levels fall off, there is less stimulation. Natural killer cells live for about 7 to 15 days. Neutrophils just only for a few days. Dendritic cells, so they all die when their function is done. And if repeated activation is there, then because of T cell exhaustion, 90% of the cells will die out. But there is active switching off of the immune system also. These two cytokines, interleukin 10 and TGF-beta, they are made by the regulatory or inhibitory T cells to switch off the immune system when the attack is over. There are other systems to switch off the immune system. One system is CTLA4. Now CTLA4 is a receptor protein which also interferes with the stimulation of T cells and switches off the T cells. This is not just all theory. One type of cancer treatment depends on inhibiting this CTLA4 so that the T cells remain alive to attack the tumor cells. Another type of inhibitory system is PD-1, which is another inhibitory system to switch off the T cells. We have antibodies like nivolumab, which is used in cancer therapy to inhibit this PD-1, to keep your T cells active to kill the cancer cells. So everything that we discussed has an application in medicine. And just the last part of it is tolerance. We saw that we have T cells and B cells capable of attacking any organic molecule. So how come they don't attack your own molecules? Because the T cells are educated in the thymus, where the cortical thymic cells, they will knock off all the T cells which have reactivity against self-antigens. Also a very interesting process where the T cells have both CD4 and CD8 markers on them initially, but they are screened, they are put through 2 examinations, and every T cell which fails the exam is killed. So only the T cells which are not going to attack your own system are allowed to thrive. This is just an overview of the functioning of the most interesting system in the body. Thank you.